Dr. Okolo, you are muted.
Good afternoon, everyone.
Good afternoon, Dr. Okolo.
Thank you. Please, I would like to share the screen from my side. It will be easier for me to manage. I hope that will be okay. Thank you.
Please go ahead.
Okay. Okay. I'll be looking at molecular epidemiology of Candida species. I'm Okolo from Jos University Teaching Hospital. Are you with me?
Yeah, we can hear you.
Okay.
Okolo, go ahead. We're with you.
Okay. All right.
Go ahead.
Okay. Candida species are typically harmless eukaryotic commensals. They can be recovered from the environment, from humans and other mammals. Only a relatively small number of clinical importance. And Candida albicans is the most frequently isolated. The typing methods in epidemiological studies are phenotypic methods and the molecular methods. Now, if we look at the phenotypic methods, these could be morphotyping methods, serotyping methods, biotyping methods, and antifungal susceptibility testing methods. What suffice to say that this afternoon we'll be looking at, briefly, the disadvantages of the phenotypic methods is such that it has very poor interlaboratory producibility. It has low sensitivity to interpersonal errors and poor discriminatory ability. The molecular typing methods are the pulse field gel electrophoresis, which is an agarose gel technique for separating large pieces of DNA. Okay. It's an agarose gel technique for separating large pieces of DNA by applying electrical current and has good reproducibility and discriminatory power. The multi-local sequence typing is unambiguous, it is portable, and a nucleotide-based technique in which different sequences in an organism is assigned distinct alleles to identify the isolates. And data generated here is suitable for global epidemiological studies. We have the deoxyribonucleic acid sequencing, which is used to determine the order of nucleotide bases in a DNA molecule, and is widely and routinely used in the identification, typing, characterization, and classification of unknown novel pathogens, usually preceded by PCR to amplify the target genes, and has very high discriminatory power and about 100% typability and good reproducibility. The molecular typing of Candida species can be for diagnostic purpose or for informative purpose. It's very useful in epidemiological and population structure analysis of Candida species. It facilitates the understanding of the dynamics of Candida species infection in human population and shows the complex relationship between commensals and infectious Candida species. It indicates the origin of an infection and enables the discovery of emergence of drug resistance in a population. It shows the genetic relatedness of isolates of the same species. Now, what are the features of an ideal molecular typing method? It must be one that is effective at discriminating between closely related but non-identical isolates of the same species. It should be able to recognize the same species amongst collection of different isolates and generate reproducible data. It must be unaffected by high-frequency genome reorganization and evolutionary pressures. It should be able to determine the genetic distance between isolates that are more closely related than those that are less so, and must be amenable to computer-based analysis to facilitate data normalization, analysis, and storage. Now, what are the biomarkers that are utilized in molecular studies with respect to Candida? We have the internal transcribed spacer region, ITS. We have the large subunit of the ribosomal RNA gene. We have the small subunits of the ribosomal RNA gene and a protein gene code for the RNA polymerase B1 gene. Now, what do we do with the sequenced target DNA? The first is we align the genes with the genes in the gene bank to identify the organism by using a basic local alignment tool. And afterward, we can edit the sequences. And there are so many softwares available now for use, from BioEdit, even the workbench tools can also be used. Afterwards, we now carry out phylogenetic analysis. Now, the computer-based analysis of DNA for epidemiological studies are such that we can use to design phylogenetic tree and also minimum spanning tree. And this requires a lot of mathematics involved. And like the minimum spanning tree requires the Kruskal and the Pin algorithm in the analysis. And also the computer-based analysis of DNA utilizes the unweighted pair group method with arithmetic averages, UPGMA, and equally the maximum parsimony and the neighbor joining methods. This is an example of the minimum spanning tree, and this is a phylogenetic tree that is unrooted and shows some percentage homology of and shows the percentage homology of the genes from the gene bank with accession numbers used for the analysis. Now, I think this may be clearer because this is almost very complex. And let's look at the next slide. Okay, from this next slide, you'll find that this is Candida tropicalis identified in a study. And if you look at T1 to T19, they are within the 40% homology, whereas T20, T21 are within the 50% homology, whereas T27, T24 are within the 90, in short, from 90% homology to 100%, we have T24, T25, T26, 27, and 28. Now, these isolates are closely related. Usually 90% is looked at as a good percentage to use in your analysis. So with a percentage of 90% homology, it tells you that these samples, these isolates, T24 to T28, are likely to come from the same source, or they are closely related, they are within 90% relationship. It tells you that if you do an epidemiological link, it may indicate either they are from the same source, or from the same geographical location, or from the same patient, or the patient, or the sources are closely related. So what percentage homology does is it gives you an idea that the isolates within 90% homology are closely related and either from the same source or from the same species. So this is used, as I said earlier, when you are doing an epidemiological study, you use samples, isolates of the same species, so that you look at their relationship with respect to their source, or with respect to the sample, or with respect to geographical location. The next slide is Candida glabrata, and it is very clear that samples isolates G4, G17, and G19 are within 90% homology. So they are very, very close and likely related. So these three isolates can be studied in depth to look at the source of the origin of the sample, and then patients from whom the samples were collected can also be looked at. If they live in the same locality, or if they live in the same household, this can also give an idea of the source and then the relationship between the isolates. So several detection and typing methods have been developed and are widely used in epidemiologic studies of infections due to Candida species. The use will depend on the cost, availability, reproducibility, and discriminatory power. This is a slide showing the training going on DNA extraction from yeast cells, and